we'll start with Trp Operon, and Trp Operon is a repressible operon. As against the inducible operon, lac operon was. This is a repressible operon. Structurally, there are 5 structural genes. There are 5 of them here. And the names of those structural genes are TRPD, TRPE, TRPD, TRPC, TRPB, and TRPA. And there is one regulatory gene also, upstream the structural genes. And that is called as TRPR. And the control elements, the control site is little bit more complicated. It's crowded. You know, it has got a promoter, it has got an operator, it has got a leader, and it has got an attenuator. I will be drawing a diagram and then you will understand. Now these 5 structural genes together, they code for 3 enzymes. So now you can understand why we say one gene, one polypeptide, because you know there are 2 enzymes which are coded by 4 genes. Okay, so therefore as a result of that, you know, these 5 structural genes, they code for only 3 enzymes, and these 3 enzymes are responsible for converting chorysmic acid to tryptophan within the bacteria. Chorysmic acid to tryptophan. You know, chorysmic acid is converted to tryptophan in with the help of some 2-3 intermediates within the cell using these 3 enzymes coded by 5 structural genes. Okay, please remember tryptophan is an essential amino acid. Okay, so bacteria faithfully, honestly indulges in endogenously synthesizing tryptophan, and as soon as the tryptophan is made available ready-made into the medium, the tryptophan synthesis, endogenous tryptophan synthesis within the bacteria stops, because when the tryptophan is ready-made available, why spend our resources in synthesizing it endogenously? Okay, but how does it happen? That we'll try and see. So this is broadly the structural features of Trp Operon. Let us see a diagram of it. So let me draw a diagram of this Trp Operon. So this is strip E, this is strip D, strip C, strip B, strip A. Some 36 base pair away, there is a transcription termination site T1, and another 250 base pair away, this is 250, and this is 36. There is another transcription termination site T2. So there are 2 transcription termination sites. T1 is around 36 base pair away from the last structural gene, the last nucleotide of the last structural gene, and then another 250 base pair away from the first transcription termination site. You have another transcription termination site T2. What is the difference? The first one is row independent. T1 is row independent. So you understand now what is meant by row dependent and what is meant by row independent. T2 is row dependent. Okay, so there are 2 transcription termination sites, and these are the 5 structural genes. So further upstream the structural region, so you have this control site, So you have a promoter. Within the promoter, there is an operator. Then there is a leader. L stands for leader. And then there is an A, attenuator. So as I said earlier, that this control site is crowded, it's complex. So you have a promoter where RNA polymerase comes and bind, and overlapping to the operator region. Means in case of lac operon, there was partial overlapping. In case of trp operon, there is a substantial overlapping between the promoter and the operator region. Okay. So promoter is the site where RNA polymerase comes and bind. Operator is the site where typically repressor protein comes and bind. In case of trp operon, these two sites are fairly significantly overlapping, right? And then these are the two additional elements. L stands for leader, which codes for a leader mRNA. L is leader sequence, which codes for a leader mRNA. It is about 162 nucleotide. And this leader mRNA is translated into a 14 nucleotide, a 14 amino acid leader peptide. Right? And then next to leader, there is a sequence called attenuator. A stands for attenuator. Right? Then further upstream with the gap, this is indicated, the gap is indicated by these two lines. Further upstream, there is a regulatory gene called TRPR with some gap. This is not immediately next to the promoter region. There is some gap. And this TRPR, it has its own promoter where RNA polymerase will bind and will transcribe this TRPR. TRPR codes for a repressor protein, which is typically 12 kilodalton monomer. And the active form is a tetramer of identical subunit. All right, so TRPR will code for a 12 kilodalton polypeptide, and 4 of them will assimilate to give you an active repressor protein, right? Now let us see this TRPE. It codes for a polypeptide, the name of which is these two polypeptides. They form an enzyme called anthranilate synthetase. I hope you understand. What does it mean? It means that anthranilate synthetase, it is a dimer. It is a heterodimer. One of the subunit is coded by TRPE and the other one subunit is coded by TRPD. And these two polypeptides, they combine to form a anthranilate synthetase enzyme. TRPC, it codes for another enzyme called indole glycerol phosphate synthetase. Single polypeptide. Indole glycerol phosphate synthetase. Again, TRPB and TRPA, these two together code for another enzyme called tryptophan synthetase. Tryptophan synthetase. Okay, so this is again an example of a heterodimeric protein. One subunit is coded by TRPB and the other subunit is coded by TRPA. So that is how these 5 genes, 5 structural genes, they code for 3 enzymes. And what is the reaction? The reaction is starting from chorismic acid. Starting from chorismic acid, So using different intermediates, the first intermediate is anthranilic acid. Second intermediate is phosphoribosyl anthranilate. Phosphoribosyl anthranilate. Third intermediate is CDRP, compound name abbreviated as CDRP. The fourth intermediate is indole glycerol phosphate. And the final product is tryptophan. All right. The first two reactions are catalyzed by anthranilate synthetase. This reaction is catalyzed by indole glycerol phosphate synthetase. And the last two reactions are catalyzed by tryptophan synthetase. So that is how chorizic acid is converted to tryptophan using the three enzymes: anthranilate synthetase, indole glycerol phosphate synthetase, and tryptophan synthetase into tryptophan. Chorizic acid using these intermediates is converted to tryptophan, right? And I told you in the earlier that TRPR, it codes for a repressor protein, which is a 12 kilodalton protein monomer, and it assimilates into a active tetramer. So tetramer is the active form. Active form, right? So this is the structural. This is the structural, you can say, structural detail. These are the structural details about the Trp Operon. So you can now understand that this is slightly more complex than the operon, lac operon that we have studied earlier. And here the control region is crowded. You know, besides the regular promoter and operator, you have a leader and you have an attenuator sequence also. And in addition, you have these two transcription termination signals. One is located about 36 base pair away from the last nucleotide of the last structural gene, and it is a row independent termination signal, transcription termination. And T2, which is located another 250 base pairs away from the first one, it is a row dependent transcription termination. All right. So let us now understand how this is regulated. So when tryptophan is not there, the condition is tryptophan is not there. Let's say tryptophan is not there. Tryptophan is absent. Okay, and as I told you, tryptophan being an essential amino acid, you know, bacteria needs to synthesize, and bacteria, nature has provided the necessary apparatus, the necessary machinery, the necessary enzymatic setup, so that bacteria can synthesize tryptophan endogenously. Okay, so when tryptophan is not there, this tetramer, let's say this is the tetramer, repressor protein. Okay, this repressor protein cannot go and bind to the operator. When tryptophan is not there, repressor protein cannot go and bind to the operator. Okay, as a result of that, this is vacant, so RNA polymerase comes and bind. So RNA polymerase will bind here, and I told you that there is a considerable overlapping between the operator and the promoter region. Okay, so RNA, when operator is vacant, RNA pol will bind over here, you know, occupying substantial space, substantial stretch of operator also. It binds primarily in the promoter region, and you know how it binds to promoter region. There are characteristic -10 and -35 consensus sequences. So with the help of that, it will identify the promoter and it will bind over there because the operator is vacant. Okay. Please understand, because of the substantial overlapping between the two, binding of repressor protein and RNA polymerase to this region is mutually exclusive. What does it mean? It means that at any given point of time, either repressor protein will bind here or RNA polymerase can bind over here. Okay. In case of lac operon also, same condition was there, though there was only partial overlapping, but binding of the two was mutually exclusive. When operator is occupied, RNA polymerase cannot bind, and when RNA polymerase is bound, the repressor protein cannot bind. So RNA polymerase binds here when tryptophan is not there, and as a result of that, you have this polycistronic messenger RNA synthesized. Polycistronic. You understand what is meant by polycistronic? Means it has got coding sequences for 5 peptides with independent, AUG, with independent AUGs, translation initiation signals, and then it will be translated to give you those 5 polypeptides. These two combine to form enzyme number 1. This is enzyme number 2, and these two combine to form enzyme number 3, and that is how chorismic acid is converted to tryptophan endogenously. Okay. Interestingly, when tryptophan comes into the medium, what you have done, just to see what happens, you have added a spoonful of tryptophan into the medium. Tryptophan you have added some tryptophan into the medium, and you are interested in seeing what happens now. So tryptophan is a co-repressor. So this repressor protein has got binding site for tryptophan. So these are the binding site for tryptophan. Okay, so tryptophan will come because tryptophan is now present, and this is tryptophan which acts as a co-repressor. Okay, so now these two together, repressor and co-repressor, binding of co-repressor will induce a conformational change. This is the repressor co-repressor complex, and conformational change means you can see the change in the shape. And this, it goes and bind to the operator region. Because of the change in conformation, it can go and bind to the operator region. And when the operator is occupied, when the operator is occupied, when the operator is occupied, RNA polymerase cannot come and bind over here. And when RNA polymerase cannot come and bind, transcription will not take place, and translation therefore will not take place. And that is what bacteria wants, because when you are making tryptophan available ready-made into the medium, what is the point in indulging in synthesis of tryptophan endogenously at the cost of its own resources, scanty resources? Okay, so that is very important. It is something like this. When you are ordering food from the hotel, what is the point in synthesizing? What is the point in making the food in-house? Okay, so something like that. When tryptophan is being made available ready-made, why synthesize tryptophan endogenously? So bacteria immediately, swiftly, showing the flexibility, it stops synthesizing tryptophan endogenously, and this is the mechanism. How does it happen? So operator site gets occupied by the repressor co-repressor complex, and as a result of that, RNA polymerase cannot bind, and transcription and translation will not take place. So the difference is, like in case of lac operon, what was happening? Lac operon is an inducible operon. What was happening? Bacteria adjusted its ability to utilize a given substrate. And what is happening in case of trp operon? Trp operon, it is a repressible operon. Okay, and bacteria is adjusting its ability to synthesize a given product, right? So this is the basic difference between the two. Lac operon, please remember, is an inducible operon, and trp operon is a repressible operon. So it remains on unless specifically turned off by the presence of the product. Trp operon, I am repeating, it remains on, it remains switched on unless it is turned off because of the presence of the last product of the enzymatic reaction, that is tryptophan. When the tryptophan comes into the medium, it gets turned off, otherwise it will remain on. Whereas lac operon, it remained off, it remained switched off, it remains turned off unless that specific substrate comes into the medium. That is, as long as lactose doesn't come into the medium, it will remain off. As soon as the lactose comes into the medium, it will get switched on. So you have to memorize this, you have to understand this, repeat it, practice it, and then you'll understand. These are two different models of gene regulation in case of bacteria. Lac operon is an inducible operon, and trp operon is a repressible operon. Tryptophan, the product, serves as a co-repressor. In case of lac operon, lactose, the substrate, acts as an inducer, right? Now let us understand a little bit more about this. In case of trp operon, the activity of enzyme and level of enzyme, or you can say synthesis of enzyme. All those three enzymes. Activity of enzyme is one thing and synthesis of enzyme is another thing. Both these are regulated by tryptophan. How does it happen? Let me just explain you. Choristmic acid gets converted to tryptophan with the help of some intermediate, right? So when there is too much of tryptophan that has been synthesized endogenously, bacteria cell doesn't want any more synthesis of tryptophan. Then this tryptophan, the final product of this pathway, inhibits the first enzyme. Inhibits the first enzyme, enthranolate synthetase. Okay, that is called as feedback inhibition. So we have already studied it in semester 1, feedback inhibition. Please remember, tryptophan is not available in the medium. Bacteria is synthesizing tryptophan endogenously, but there is too much of tryptophan that has been synthesized, and bacteria right now doesn't want any more synthesis of tryptophan. In that case, tryptophan, the end product of the pathway, it goes and inhibits the first enzyme of the pathway, that is enthranolate synthetase, by the mechanism called as feedback mechanism. Okay, and that is how the synthesis, endogenous synthesis of tryptophan will be regulated by regulating the activity of the enzyme. Okay, this is activity of enzyme. Second is synthesis of enzyme. You know, bacteria is involved in the synthesis of tryptophan endogenously, right? But when tryptophan has been made available ready-made into the medium, then this tryptophan acts as a co-repressor. It binds with the repressor molecule and goes and occupies the operator region, not allowing RNA polymerase to bind to the promoter. And therefore, the transcription of the structural gene will not take place. And synthesis of those enzymes, those three enzymes, which are responsible for the synthesis of tryptophan endogenously, it will stop. So, in case of Trp Operon, both, please understand the activity of the enzyme is regulated by feedback mechanism by the tryptophan molecule, and synthesis of enzyme is again regulated by the availability or non-availability of the tryptophan into the medium. Okay, this is another thing, right? We'll take some examples of what we studied in the lac operon also. Okay, so let's say if there is a mutation in TRPC, what will happen? If there is a mutation in TRPC, what will happen? So only TRPC gene and its product. What is its product? Its product is enzyme indole glycerol phosphate synthetase. Indole glycerol phosphate synthetase. This is going to be affected. TRPC and its product, that is indole glycerol phosphate synthetase, this only is going to be affected. Okay, and it again depends on whether mutation is taking place at what place. So depending upon the site of mutation, this may be harmful or this may not be harmful. So the point is only that particular structural gene and its expression and its product, they are going to be affected. But if there is a mutation in regulatory gene, that is TRPR, what is going to happen? This entire structural genes, all the structural genes, their expression is going to be affected irrespective of the fact whether tryptophan is present or tryptophan is absent. That means the operon is going to be constitutively expressed. We want only the operon to be expressed only when tryptophan is not there. But if there is a mutation in TRPR, irrespective whether there is a tryptophan available or tryptophan not available, the entire operon is going to be expressed. That means all the 5 genes are going to be expressed. That is called as constitutive expression. So you understand what is the difference between a regulatory gene, mutation in regulatory gene, and mutation in a structural gene. It happened yesterday when there were some connectivity issues at my end. Okay, so when I lost the connectivity, you know, all of you could not attend the class. So in our relationship, I am the regulatory gene. For example, now initially there were 21 students who joined, but now there are only 16 of them available. What does it mean? They lost either connectivity or there are some other technical issues. So if they lost connectivity, of a student who loses connectivity, only he or she gets out of the class. All remaining others, they're going to be there. So this is the kind of thing. Okay. Now let's say there is a mutation in promoter. There is a mutation in promoter. So promoter is what kind of site? Promoter is a cis-acting site. What is going to happen if there is a mutation? RNA polymerase will not come and bind, and as a result of that, the transcription will not take place. Okay, and this situation, original situation, cannot be restored by adding another promoter, because promoter is a cis acting site. Okay, if there is a mutation in TRPR, that is what we have seen over here, it is going to be constitutively expressed. But suppose if you make it partially diploid by adding a TRPR plus a wild type TRPR, then what is going to happen? The original situation, wild type thing is going to be restored, because TRPR is a cis acting thing. TRPR is a trans acting thing. TRPR is a trans-acting site, or TRPR is a trans-acting gene. What does it mean? That this TRPR is going to synthesize a normal repressor protein. This normal repressor, if tryptophan is available, will combine with it, will form a repressor co-repressor complex, and this repressor co-repressor complex will go and bind to the operator when tryptophan is present. So in presence of tryptophan, this operon will get switched off. Otherwise, when TRPR is mutant, irrespective of its presence or absence, this was going to be continuously expressed, that means constitutively expressed. You can take similar examples when there is a mutation in T1 and there is a mutation in T2. What is going to happen? This T1 and T2 sites are also cis-acting sites. All right. So tomorrow we'll understand sorry, another mechanism of regulation in case of Trp Operon, that is called as attenuation control. So the meeting will end anytime soon. Thank you very much for attending the class.